my next guest, who has been looking at so many different aspects of this from a super interesting perspective, but probably the most interesting is the fact that my next guest, Dr. Robert Malone, claims that he is the inventor of mRNA technology. That's right, the technology that's being used in the Pfizer vaccine, in the Moderna vaccine. And so if we're going to have questions, perhaps some answers can come from the person that was at the heart of this. I am so excited to be joined right now by Dr. Robert Malone. Dr. Malone, first of all, thank you for taking the time to join us here on the High Wire.
My pleasure, Del, and thank you for the opportunity to speak to your broad global audience.
Absolutely. So let's just start with the first question. You're doing a lot of interviews right now. There's one video that's about 3.5 hours long that's getting a lot of traction, but I have a lot of questions, so I want to get to it. First of all, you are the inventor of mRNA technology. I suppose because everybody else has ever come on this show, someone out there is refuting what they say, even though they have, all these letters behind their name. Tell me, how is it that you make that claim of being an inventor of mRNA?
Thanks. Yeah, so what's going on is that a number of people think that there's a Nobel Prize in play, and that may be the case. And in my experience, when that kind of thing happens, it brings out certain characteristics in people. People get very competitive. It's something worth having. It's, a form of immortality if you get that. For me, fortunately, I kind of cut my teeth at a place called the Salk Institute, created on the cliffs of La Jolla, looking out over the Pacific by Jonas Salk.
Yeah.
It's kind of a temple to vaccines. And at the time when I was there was about a half a dozen Nobel laureates, including Francis Crick. So for me, I've kind of seen what, at a very young age, at 28, I've seen what this can bring to people, how it can disrupt their lives and their family lives and everything else. And I don't know that it certainly would be a lovely thing, but it's not my life goal. I'm as I said, I was a farmer in a ranch hand before I was a physician and a scientist, and family and my farm and my horses matter a lot to me. That's kind of where I'm centered. So be that as it may, it is bringing out some people that are trying really hard and lobbying very aggressively. And behind them, in particular, is an institution, University of Pennsylvania, that holds a patent that's being used by Moderna and also by BioNTech. And she's an employee of BioNTech right now, a vice president, as I understand it. And so both herself, Drew Weissman, and Penn stand a lot to gain should she win this prize. And so what's happened from my point of view is I've kind of been written out of history. There's a lot of failure to cite in academic publications and on Wikipedia. You can look it up. It's all about Katie. And I've been reassured by colleagues at the Karolinska that have been on the committee in the past and are very aware of the process, that I really don't need to worry too much about that they have a very professional due diligence team. But every time there's people that think that if they can push the press and get a journalist to champion their case, then that'll improve their ability to do it. No, so that's just the way people are.
Okay.
And it's there it is. In terms of what happened.
So what happened?
It's a super story. I love to tell it to graduate students and others. It's a lot of interesting little parables buried within it. I was originally a big fan of retroviruses, and that was my core background. I was at a lab at UC Davis that really had the very first indications that a retrovirus was associated with an AIDS-like syndrome in monkeys. And at the time, I was working on mouse mammary tumor virus, another retrovirus that causes breast cancer in mice. And so as I moved into my MD PhD program, I thought that the thing that I could do that would just make a lot of sense and would make for a great career was to focus on gene therapy using retroviruses. And at the time, the two top labs in the world were both in San Diego. One was Ted Friedman's lab. He was on the main campus, and he was the guy that originally came up with the idea in the 70s. And the other was a brilliant scientist named Inder Verma, who had been trained by David Baltimore, who also got a Nobel Prize. And Inder was championing and really driving forward both the use of retroviral vectors and adenoviral vectors for gene therapy purposes. So as a young graduate student, even though I was warned not to, very ambitious, I went to this postdoctoral research lab at the Salk Institute under Inder in the molecular biology and virology labs. And I wanted to work on some nuances. You know, as a graduate student, you have to figure out what you're going to get your thesis in. And I wanted to work on how RNA gets assembled into retroviral particles. Basic science question. And so in order to do that, I thought, well, what I really needed to do was figure out some technology that would allow me to synthesize large quantities of very pure RNA, put it into cells, and get those cells to put it back out, package it as a retrovirus particle. So that was the whole idea. I was, banging away at this, trying to come up with technology. And there was a series of kind of stepwise improvements. And a lot of this had to do with being in the right place at the right time, surrounded by geniuses, and access to amazing information and technology. But it comes down to about four major events. One of them was I was being kind of mentored by a postdoc named Dan St. Louis in the lab, who was working on a system involving putting genes into cells of a mouse in cell culture, and then taking those cells, implanting them into the mouse, and watching for how much protein and how long this gene therapy protein would be expressed. To his great frustration, after about 3 weeks, the protein stopped being produced in the mouse. It was a great kerfuffle. Everybody thought there was a sophisticated mechanisms involving gene silencing. And as the half-trained MD that I was, I said, this looks like an immune response because it's the perfect timing. At the time, that was kind of heresy because no one had really grappled with what would happen with gene therapy in terms of the immune system. Now we all recognize it as one of the major problems in gene therapy is if you put a foreign protein, if you express a foreign protein in your body, and if you have cystic fibrosis or muscular dystrophy or whatever the thing is, your body, your immune system doesn't know that it's the right protein. It doesn't know that it's a good protein. It only knows that it's a different protein that it has, and it'll start attacking it, and it'll attack the cells that are making it. So, it's this simple. I run into this problem. I am passionate about gene therapy. I think it's my whole rest of my career, and then bam, I hit the wall. What am I going to do? So this is one of those make lemonade out of lemons moments. Because I'd come out of this lab in Davis that was focused on developing vaccines for AIDS, it's no surprise, no huge leap for me to say, oh, I know, we're gonna make vaccines using gene therapy. We're not gonna use it for gene therapy because that's gonna be kind of pretty hard, but we can use it for vaccines. Now, interesting aside, there's the senior postdoc in the lab is a man named Dinko Valerio. Dinko left the lab and founded a company called Crucell, and it was focused on adenoviral vector gene therapy. And he came to me at a scientific meeting a few years later, and he said, Robert, you're right. I have to change my company's focus and pivot to focusing on making vaccines instead of gene therapy using adenovirus. Crucell was then exploded in growth. It was sold to Johnson & Johnson, and that gives rise to the J&J vaccine. Okay, so
Wow.
with the RNA story, so this is the first of the big ahas is, we got a problem with gene therapy, immune response, but we can turn it around, make lemonade out of lemons, and use gene therapy technologies of a variety of sources to make vaccines.
Just
Next
explain
step,
to me for a layperson, why vaccines? Like, what is it about a vaccine that technology you just immediately thought, oh, it worked great for that? I mean, I get why it's bad for gene therapy because it gets attacked, it can't keep reproducing, whatever it is you're trying to achieve, it gets killed off. But why did you say, well, this would work for vaccines? What is it about it
two
that makes it good?
two reasons. Two reasons. First reason is that with almost all of these gene therapy technologies, including the earlier RNA versions and to some extent the current one, the levels of protein produced, in other words, the efficiency of the gene therapy is actually quite low. So the idea that we could give somebody an injection and it would go to all the cells in their body is pure science fiction. You get very small numbers of cells that get the gene, and they don't tend to produce a whole lot of protein in their, once they kind of get subverted to be little protein manufacturing factories in your body. And so the whole big idea that Ted Friedman had that we were going to cure all these pediatric diseases obviously hasn't come to pass.
Right.
And so I knew that vaccines, the thing about vaccines is you don't need very much protein. A little bit will do the job. So that's point number one,
So a little
is
bit is
in the
all you
spectrum
need
of,
to make the
yes, a
antibodies
little
and all
bit's
that
all you need.
production to fight it, and your body develops an immunity, and that's all you needed.
A little bit is all you need, and that's all you're going to get
Understood.
for the most
Okay.
part. So, make the best of it. And then the other thing is that traditional vaccines often, you put the protein in your arm, you get the jab, and it's mixed with something that makes the immune system react to it. We call it an adjuvant.
Yep.
And it kind of stays put, and it doesn't go into cells. And all the complex, we could go into the immunology, it's very complicated and wonderful. But the bottom line is that those kinds of traditional vaccines tend to be biased towards producing antibodies. But if you want to control a virus or you want to kill a cancer cell, antibodies can do that using antibody-dependent cellular cytotoxicity, big fancy science word. But the immune system also has another branch, which is cells that learn to directly attack other cells. We call those cytotoxic T lymphocytes.
Okay.
Another big fancy sciencey word.
Yep.
But in order to get both antibody and CTL, that's the acronym for cytotoxic T lymphocytes, it's really good if you can actually make the protein in a cell of your body.
Okay.
And then your immune system reacts to it as if it's infected by that virus, but there's no virus, for example, okay?
Yeah.
Or if you're a cancer cell and you're expressing a new protein that's mutated somehow, you'd like to be able to have a cell that would be able to go and find that cancer cell that is expressing that mutated protein and kill that one, but not kill all the other cells. Kind of
Right.
important.
Yeah.
So CTLs are really good, and they're probably why cancer is so rare in the first place. There's a good chance that the odds are that we all get cancer in terms of one or two cells at various times in our lives, but our body's immune system is clearing it for the most part. It's only rarely that it gets out of control. So CTL. So the idea about gene therapy based vaccines is that it can elicit, that strategy can turn on both antibody production and cytotoxic T lymphocyte production. And it kind of makes lemonade out of lemons because unfortunately, we're not that good with most of our gene therapy technologies. And we have failed to cure cystic fibrosis, but it looks like we can do a pretty good job making immune responses,
Okay.
whether it's an adenoviral vector or a mRNA or other platforms for expressing these things. Does that make
Can we
sense?
say that both adenovirus vector and mRNA are gene therapy technologies? I mean, we hear that and
Explicitly.
throw it together. Are they the same?
Yeah.
Okay.
It's a messaging thing that the public health service doesn't like because they think that'll turn people off. But I'm a scientist. I live in the world of evidence-based medicine and data and science, and I kind of my brand is I call things as I see them.
Right.
And as I've explained, this all comes from a single gene therapy lab at the Temple for Vaccine Development, created by Jonas Salk, on
Right.
the banks of the cliffs in La Jolla. I don't know how to say it any more clear.
I
You
think that's
have
pretty
genetic
clear.
material, you're putting it in a cell, you're expressing a protein for a vaccine purpose. It's gene therapy applied to vaccines. That's what it is.
Okay, just so I have
You
a
can
sense of
dance around that.
timeline, what year is this all like? Are you making these connections and said, hey, let's make it a vaccine since it doesn't work as a gene therapy? How long ago was that taking place?
1987, 1988, and 1989 is
Okay,
when all these patents were filed, and there's numerous patents. Yeah,
okay,
this is the thank you. This is the base uh, PNAS article that describes the large scale manufacturing of mRNA, its purification, its delivery by coating it with another fancy sciencey term, lipids. You would
okay,
call it fats, perhaps.
yeah,
Um, and then there's this one from Science, and this was a interesting story. So there's like four major discoveries that happen that get us to the base patents, and this is the last of those four. And that also is a fascinating story.
okay,
We were I had previously shown that, as you showed in the first paper, that we could put fats around RNA, and if it had the right structure, and I showed what those structural elements, they still all use that same stuff, the same manufacturing process basically, but bigger, the same structure of the RNA, etc. Okay. Um, in that paper, uh, I showed that that technology worked for a huge range of cell lines. Um, and then because I was teaching embryology and I had extra tadpoles that I'd prepared. Okay, these are African clawed frog tadpoles to teach
okay,
undergrads. Okay, I had extras, and I just said, well, let's see what happens if I put this mixture that I'm using on the cells on these tadpoles. And lo and behold, the tadpoles express the foreign protein also. So then I did a similar thing with DNA and chick embryos because I was working with both DNA and RNA, and those also express the foreign protein. Now we've got something interesting.
so now
And
we're
at
doing
that
what your
point,
dream
they're
is to have the cell itself expressing.
totally unexpected.
Okay,
No retrovirus, right? It's just totally amazing that suddenly I find myself at the birth of non-viral gene therapy. Totally unintentional, but it
wow.
just, chance favors the prepared mind. Um, so it happened, and you're surrounded by intellectual giants and all this technology and ideas, and you're a young, uh, overly aggressive, not very humble scientist trying to make your way, and things happen. Um, so, and things did happen. Uh, a bunch of fights started taking place, and I got caught in the crossfire. I wasn't prepared for it. Patent fights over who's going to be the inventor, and does UC San Diego own the patent? Does a Salk own the patent? UC San Diego tells me I can't talk to the Salk. Salk tells me I can't talk. It's just crazy, and
Wow.
and everybody smells money, and I am just a tiny little speck, right? I'm just a graduate student, working for the guy that characterized reverse transcriptase for David Baltimore. He's a really big shot. Um, and we don't need to go into Ender, but you can
Right.
look up other stuff about Ender.
Okay.
Um, so I just I I kind of have a meltdown. I end up with post-traumatic stress disorder. I have to leave the lab. I just psychologically can't stay there. And my wife has to finish her bachelor's degree, so I need someplace to park myself before I go and finish my MD. The guy that I've been collaborating with at a company in the Bay Area called Syntex has just quit Syntex and joined a tiny little startup in La Jolla called Vical. And there he's supposed to be working with Doug Richman and Carl Hostetler on development of new drugs for AIDS and liposome encapsulation. And he says, hey, Robert, you want to leave the Salk. Why don't you come over and work for me, and we will set you up a little skunk works. And in parallel, he sets up a collaboration with a guy named John Wolfe that's just left Ted Friedman's lab at San Diego and gone to the University of Wisconsin. And Vical doesn't have any animal facilities. John Wolfe is given access to animal facilities at Wisconsin, And so we set up a deal where I come over, I bring all my reagents, my plasmids, technology protocols, blah, 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 and come over to Vical, start work as a technician, and manufacture these things and ship them off to John Wolfe and his team for injection into first rats and then mice. And this comes on the heels of the findings that it works in frog embryos and chick embryos, the frog embryos disclosed in the PNAS paper.
Yeah.
So we send John Wolfe the samples, and lo and behold, it works. We're all ecstatic. Holy, hallelujah.
Right.
But there's a naysayer. There's a chemist in the group who's making some of these cationic lipids who just insists that we're not doing good science. His name is Raj Kumar. He ends up running a transmission shop eventually in San Diego. That's a weird part of the story. But Raj is the chemist, and Raj insisted that we're not doing good science. And the truth is, he's right. He says, you're not doing all the negative controls. And so to placate Raj, who I'm sure is absolutely positive is going to be wrong, I include samples that just have the lipids and just have the RNA alone and ship them off to John Wolfe for injection. The data
two
comes
separate
back.
ones. One just has lipids, the other one has just the RNA, not combined.
And then other samples that have both.
Okay.
So the negative controls and the positive controls.
Got it.
We already know that they're working. Okay. Because Raj Kumar was a jerk and insisted that we weren't doing the right science. Okay.
Yeah.
And I'm being facetious a little bit, but setting up the story because you can figure out what the punchline is already. So John's graduate postdoc does this experiment, injects it into mice, runs the assays, sends us a fax with the data. John's ecstatic. It worked again. And I say, yeah, John, but the negative control is positive. Matter of fact, it's even more positive than the positives. What's going on? He says, my post, precisely, that is the study. Thank you. So the postdoc repeats it. And this is actually the repetition. And lo and behold, the RNA alone is working. And then that starts a whole nother cascade. And with that, this is just the lab notes from synthesizing the RNA. Everybody says RNA is not stable. That's, yeah, in some cases, but if you're good at how you prepare it, and obviously we can make stable RNA now in large quantities. At the time, it was another one of those heresy things. Right. Everybody knew it's another group think, just like we're facing with this outbreak. Only on a smaller scale, everybody knew that RNA was unstable, except for those of us that were working with it and synthesizing it. So there it goes. So Raj Kumar was right. And then suddenly we had naked RNA delivery. It didn't have to have the lipids necessarily. And we tried it with DNA, and it worked with DNA. And then another whole slew of patents start getting written. And I'm off with the lawyers disclosing this, that, and the other thing, and cooking up all these crazy, wild ideas about how we could use this for vaccines and other applications. And that results in, 30 worldwide patents that are issued that all have the same filing date. And by the way, a patent gets filed from the Salk from my prior stuff. It all gets filed on the same date because Inder is consulting for Vical. And that's a whole nother part of the story we don't need to go into.
Okay.
But that's how it all started, was this series of events. Like I said, chance favors the prepared mind, and being on the shoulders of giants at the right place at the right time in the temple of vaccinology. That's where it all started, and that's how it happened.
So let's do this. I'm sure this story goes on in your career and how we get here, but let's jump a giant part here because there's a bunch of things I want to ask you about. Let's cut to, say, somewhere around 2019, end of 2019. This pandemic breaks out. I'm not going to get too big in the details of the pandemic. We've had a lot of specialists get into whether or not, how deadly this virus is, all of those questions. I want to stick with, at the moment we start hearing about early 2020, that we are going to be able to rush what appears to be a brand new technology. It's discussed as being a brand new technology by Moderna. We know that NIH, Tony Fauci, seems to have some affiliation with the Moderna patents. Some of his scientists are directly involved with patents involved in the Moderna technology, Pfizer, and we see what this warp speed by at that time President Donald Trump. We're going to warp speed this vaccine process. Now, you don't know who I am. I doubt you've done a whole lot of research. So just very quickly, from the very beginning, we were very alarmed at that issue. I've been looking at vaccines and vaccine development for years. It's part of where I've been focused as a medical journalist. And one of the things that I said on this show as soon as we heard about this is there's no way to warp speed a vaccine technology. You may be able to warp speed the technology, but you can't warp speed a safety trial. A safety trial has got to last years. We don't have any magic way of in 3 weeks or 6 months determining what happens to you 3 or 4 years down the road. We need a 3 or 4 year trial to see that's taking place. So that's where I was coming from. I've been attacked by New York Times. I've been attacked by newspapers everywhere saying, you're spreading misinformation. You're causing, vaccine hesitancy. I said, I'm not causing anything. We are trying to demand that proper safety trials be done. I also want to point out that our nonprofit, the Informed Consent Action Network, we have won lawsuits against the FDA, against the National Institute of Health, against the CDC and Health and Human Services, all based on information that they weren't sharing properly with the population about vaccines, around liability, all these other issues. But because of that relationship our nonprofit has, we reached out and had a petition to the FDA in the beginning of the phase 3 vaccine trials, and we demanded that they add a saline placebo group to the phase 3 trials, or we would come out against those trials saying they weren't proper safety trials. A safety trial against a meningitis vaccine only tells you it is or is not as safe as a meningitis vaccine. We wanted a baseline safety based on a saline injection, and they changed the phase 3 trials about a week after we petitioned. We feel like we had a large part to do with that. So that's just giving you some background of our interactions with the FDA, things that we care about. I'm just trying to make sure that products that are used by people are safe. I'm just a consumer advocacy group, really. And then I also believe we should always have choice. That's our number one issue. I believe in informed consent. I think you have the right to opt in or opt out. So that's very quickly sort of my last 5 years of investigation. The show we've been using to detail the lawsuit wins and things we've been discovering in our investigation for about the last 3.5 years this show's been here. So all that being said, from your perspective, this is a technology that you understand. I don't know how attached to it you still are, but we suddenly, mRNA is now all over the headlines. They're going to warp speed this vaccine into production through safety trials. They are literally saying to us on television, we are going to start manufacturing hundreds of millions of these vaccines as they're starting about phase 2, I think, trials, not even phase 3, just if it works, we want to have it ready to go. What were you thinking about that moment? Or if there's a more particular moment before that, let me know. But since this is a technology that you were there at the beginning, you gave birth to this thing, and now the baby is about to be released on the planet Earth, what were you thinking?
So I want to start off by applauding the story you just shared. It sounds like you and I are remarkably aligned. I'm a physician and a scientist, and I'm rigorously trained in bioethics. And I've written about the bioethics of experimental COVID-19 vaccines and what I see as major issues that I'm concerned about. So you ask about so I'm going to go back to beginning of 2020.
Okay.
As I mentioned in the preamble before we started recording, I've been at the tip of the spear for too many outbreaks now: flu and Zika and Ebola and AIDS, etc. So
Well, let's just
I'm
cover
seasoned.
why. So, because you and I had this conversation, I had asked
Yeah,
you in one of your interviews, you talked about working for DoD, Department of Defense. So I'll let you sort of,
yeah.
yeah.
So, so I get a call from a intelligence person who happens to be in Wuhan in December of 2019. Let's not go there. He calls me in the first week of 2020, January, and he says, "Robert, you got to get going. We've got a problem with this virus." Shortly thereafter, the first viral sequence is disclosed, and I get my team spun up, and we start using advanced computational technology to try to screen drugs for potential activity. That's the thread we'll talk about later. Um, at the time, I'm active on LinkedIn. I avoid Facebook, and I rarely interact on Twitter, although lately I have been particularly after the podcast.
Mhm.
Um, and I make a threat assessment, as I have done in previous outbreaks. And my assessment is that to do, as you say, to develop vaccines for this pathogen in this time frame, particularly given the long and rich history of failed vaccine attempts due to antibody-dependent enhancement for coronavirus vaccines, veterinary and human, although there are a couple of licensed veterinary coronavirus vaccines that have overcome the problem. Um, my assessment is to do this responsibly um, for a vaccine is going to be too time consuming and the threat is too present. And people can go back, I guess some people do, as they Google my name and various topic areas, and my old LinkedIn posts pop up. I've been blogging all the way through this. On the vaccine front, I've been very clear about my concerns about ADE and other complications, and um, what I perceived, just as you did, to be risks in the strategy that the government was taking.
Let me just insert
And
very quickly for some of my audience that may not know what ADE is. My layman's perspective is we saw in animal trials with coronavirus vaccines for over a decade now that when they would give a vaccine for coronavirus, many different attempts at it, they would put it in animal ferrets, sometimes cats. They would give the animal the vaccine. It looked like they were creating robust antibodies. It seemed safe because the animals weren't dying. But it was in the challenge study, something we don't usually do in human trials, where we challenge the animal with the coronavirus by injecting the coronavirus. We saw something happen which was somewhat shocking. Instead of the antibodies actually protecting the animal, it seemed to help the virus proliferate through the cells, turn into what was called a cytokine storm, which is sort of an immune system meltdown. Lots of problems, Th2 immunopathology in the lungs, things like that. So upper respiratory issues. We've seen this in the RSV vaccine program back in the 1960s.
right,
A vaccine did this to children. We discontinued that. There's some belief that the dengue vaccine, Dengvaxia, that was given just over a year
yep,
ago in the Philippines had the same issue. Over 1,000 people are said to have died from that vaccine, and they kicked that drug company and that health minister out of the country. That's just a quick, for anyone that's brand new to the high wire, this is something we've reported a lot on, and it was one of our major concerns with rushing this vaccine. If you have a vaccine that helps the virus kill the host or helps the virus, proliferate and cause serious disease, then the vaccine is going to do the opposite of what you want and could be detrimental to your species if you plan on giving this to every single person alive. That's my sum up. If you want
so
to correct any of that, but
well done.
okay.
You're really up on this, and what a pleasure to have an interview with somebody that's so familiar with the topic area and has taken a brave stance
Yeah.
through time. So one of the things that I pointed out that we probably also share, or maybe not, I don't know, um, is a belief that rushing this in the way that it's been rushed, and I know they put out graphics that say things like uh, um, uh, "We didn't cut corners, we just straightened out the road," I think is one graphic that
Right.
the U.S. Public Health Service has put out. Um, but but anybody that can do simple math can see that normally we have to have at least 2 years of safety data after your 3,000 subjects in your phase 3 total have voluntarily received the product, and we follow them for at least 2 years and look at all those safety data to look for long-term things like autoimmunity. And
Mhm.
we can do the math. and the vaccine has not been jabbed in people's arms for 2 years, let alone, right? So
Yeah.
that's just the most obvious example of them cutting corners. My point was back in the day when you were thinking about this and I was thinking about it and blogging on LinkedIn about it, is that we face the risk that we basically validate the criticisms that have been mounted over the years by people that are labeled as anti-vaxxers.
Hmm.
Um, this willingness to suspend the rules and the normal processes and compromise key safety guardrails that have been developed for decades, over decades, as the way that one needs to safely develop vaccines that cause us to require a decade before we typically will license a vaccine. If we circumvent those, we validate the whole meme
Hmm.
that pharmaceutical companies and the government are willing to play fast and loose with people's safety. And I was worried back then that this blind rush was going to put us in a position where the legitimacy of the public health officials structure would be legitimately called into question. And lo and behold, here we are.
And just to
And
sort of say we are that aligned, I've reported, I think 3 times I've been a speaker in public comments during the ACIP meetings on this vaccine. I said exactly that. If you push this through, you're going to destroy the credibility of the CDC. You will destroy the credibility potentially of our trust of health departments and our doctors. You are putting at risk our belief in our national healthcare system and the healthcare system of the world.
the whole vaccine enterprise.
Yeah.
You're validating the concerns that people have been voicing about the fundamental pediatric vaccines.
Yeah.
Um, we are completely aligned. This is
Okay.
bad public policy.
Agreed.
I'm not afraid to say that. Uh, so yes, we have been aligned, uh, and yes, I have. So please understand, I'm not somebody who seeks media attention.
Right.
I've dealt with the media for decades. I'm almost always on background, trying to help journalists to understand the science and the issues so they get it right. Because we all, I think, as scientists and physicians, have an interest in having the media and mainstream media accurately communicate scientific issues. So
Hmm.
I kind of think it's an obligation to do what you can to help the media understand topic areas and issues in science and medicine, and I have done my best. But I'm not somebody who seeks to be on CNN or whatever.
Okay.
Um, it's
That being
it's kind
said,
of
what were
with
the alarms? Let's get to what are the red flags? This thing's being rushed
yeah.
out.
So
Someone that designed this, what were you concerned about?
so um, as we discussed earlier, I have, as part of my work, my business, I have network of colleagues and friends that I trust, and they trust me throughout the government because a lot of what I do is government work, and that includes people in senior positions at the Food and Drug Administration.
Okay.
And for quite a while, a small group of us were meeting every other week on Fridays and having a Zoom call and going over issues. And as this came to fore with the spike, and I was very aware of the biology of spike because it relates to one of the repurposed drugs that we're working with, and I told them this isn't just an antigen. This is a biologically active protein, and it has effects like modulating immune response and other things. And it's being treated as if it's just an antigen, and it's not. These viruses pack all kinds of functional properties into their proteins and their genes because they have to have a very small, compact genome. It's just the nature of RNA virus biology. So spike isn't just a antigen; it's a biologically active protein that does more than just bind ACE2, the receptor that is
Yeah.
used by the virus to infect cells. And so I alerted them and I said, "Hey guys, are you aware of the implications of what's being done here. And I sent them the manuscripts, and my colleagues were sufficiently aware and cognizant of the argument. And so they forwarded it to others within the agency, within the Center for Biologic Evaluation and
What would be the
Research. Those are the
timeline right
people
now we're talking
responsible.
about? When was this that you had made this determination?
September last year.
So September of last year, and just to be clear again, just for my audience, what we're talking about is the spike protein, which is just one of the proteins on the surface of this virus. It happens to be what grabs onto the ACE2 or the ACE2. So the idea being, if we target a vaccine to just go after that instead of the whole virus, the usual way we make a virus, let's go after the weakest part of the virus maybe, and then if we kill that or stop that ability to connect, then we can sort of harness and stop this virus. But the problem you're talking about that you are expressing, if I'm correct, is that the spike protein, which we now are seeing, has its own properties of being toxic and dangerous. So therefore, it's not just like a key that they thought sort of unlocks a door. It actually is a weapon and has its own effect on the body. And if we're making a vaccine just around this spike protein, we're going to send a toxic element, not just an antigen, not just something that's somewhat harmless and all it does is open up the ACE2. We're talking about something that could very well be what's leading to the blood clots, all the strokes and the issues that we're seeing.
Yeah, and the data are increasingly supporting that.
But
So
you said this all the way
the
back in September. I want to make that clear to everyone. The
yeah,
FDA then is aware of this. Okay.
and I kind of they blew me off. They said we don't see that as a problem, and we're going to proceed, and we're not going to worry about it. Basically.
Okay.
So they didn't think that the documentation around that was sufficient. Now, since I sent in that with some of the papers that we were focused on, additional work came out. So there was a study with cultured human brain-like structures that we call organoids. This is the kind of the brave new world from stem cells and everything else that we're using so that we don't have to burn so many mice, and so that we can model things that are more like humans without having to test them in humans.
Mm-hmm.
So brain organoid study, peer-reviewed, showed that spike opens blood-brain barrier in that model. Then there was a mouse model that shows that it opens blood-brain barrier. Then there were other studies that confirmed that. Then there was the press release from our friends at the Salk Institute with data that showed that spike is directly cytotoxic. Now, I've been fact-checked by PolitiFact and our friends at Reuters,
Well,
and
we
they
have that in
both said
common
that this is false. This is false information I'm giving out, and they gaslighted me, good and hard. But fortunately, I'm a scientist that doesn't say things just out of the blue because I feel like it or I think that it's the right thing to say. I say things because there's data behind it. And so I was immediately able to flip the multiple peer-reviewed publications and call them out. Of course, that in the modern world, being mainstream media means you never have to say I'm sorry or retract anything.
So just very
it
quickly,
was,
why would it matter for people watching? Why does it matter that the spike gets through the blood-brain barrier? Why isn't that what we want it to do?
It's not just that it gets through it; it's that it opens it. So what he's talking about, or we're talking about the blood-brain barrier, you can think of as kind of a bunch of little interdigitating fingers between cells, tight junctions, and structures behind them that function as kind of a filter. And the brain is a special place. I think most humans can agree with that. And we really don't want to have bad stuff getting in there because it's kind of what makes us human. So opening up the blood-brain barrier means that things that are in your blood, like say other viruses, the toxins, all kinds of stuff that normally is sequestered so it can never get into the brain compartment, it suddenly is able to do that. That's bad, and it can cause inflammation and all kinds of things. And the fluid shifts that occur can cause changes in mental status, potentially can cause things like brain fog. As a personal long hauler who's survived COVID, I know brain fog well. In fact, some people that receive vaccine complain of brain fog after vaccination. I can't say how often because the databases are not good, but it happens. So there's a lot of, some people get after vaccination neuropathy. that's numbness or other things. And so spike, the things that we know that native spike does seem to overlap with some of the symptoms both of COVID and of this post-COVID genetic vaccine syndrome that some people report.
Mm-hmm.
So this is one of those, it looks like a duck, walks like a duck, quacks like a duck, but I can't say it's a duck. However, and I'm criticized for saying that this could be happening with the vaccines because many people assert that the NIH and others that engineered these spikes for use in these vaccines engineered them so that they would be safe and they wouldn't be toxic. Well, that's a little bit circular because at the time they were denying that they were toxic.
Right.
So why would they engineer them to be safe? What they did was they engineered spikes so that it's locked into an open conformation, which is what it uses to bind ACE2. They also put a tag on it so it stays put in the cell that is expressing it, transmembrane domain. So these were the genetic modifications that were made, but they weren't made, I don't think, for safety purposes, at least the locked in open conformation. That's to make sure that the receptor binding domain stays open so that you can better generate antibody responses and educate B cells to make antibodies that'll stick into that groove. You don't want it to vary between the open conformation and the closed conformation that's used for cell fusion, because that can interfere with the ability of antibody education, B cell education, to generate antibodies that recognize that receptor binding domain.
Okay.
So I believe that's why they did it, not for safety purposes. But the argument is made that one cannot assert that just because spike is toxic, that the expressed spike is also toxic. My counter to that is the way the rules work with drug development is kind of like the French judicial system. You're presumed guilty until proven innocent. Okay, and it makes a lot of sense that we start with the assumption that anything new, particularly anything genetically engineered, is not safe. And it's the obligation of the drug development company interacting with the FDA to prove that it is safe, not the contrary. Okay, and so I hear, I'm active on social media, and so I get all of the criticisms, as you probably do in your blogs. And everybody's a genius now and an expert, and they don't hesitate to tell me where I'm, that I should pound sand. And this is often the criticism is I can't assert that the vectored spike is similarly toxic to the proven toxicity, cytotoxicity, and activities of native spike. And as I mentioned, my retort to that is, well, that's fine. Show me the papers where the pharmaceutical company proved it, and I'll shut up. But I have not seen those data, and I'm not sure they exist, and I certainly haven't seen them in peer-reviewed publications. So there it is. I kind of fall into the camp guilty until proven innocent, and it could well be that the modifications I speculate that lock the receptor binding domain open in spike could actually make it what we would call a super agonist, something that would bind even more efficiently than the unlocked spike. I don't know. It could well be that it's more toxic than regular spike. Prove it to me. It could go either way. It's science. Let's do it. Let's see the studies. Show me the data.
And you're right. And I was in an interview by 4 Channel or whatever, Channel 4 out of England, and the reporter who, they used to say I was just, a liar and a hack. Now they say I'm just cherry picking my information. When she said to me, what I dislike about what you do, Del, more than anything is you are going by the assumption that the vaccine is dangerous just because it hasn't finished its safety trials. You make the assumption it's dangerous instead of making, and which scares people, instead of making the assumption that it's safe. And I said, oh, you know what? There's actually a term for that. It's called the scientific method. It is that we are supposed to challenge the theory and challenge a product and believe first and foremost that it is dangerous and show all the reasons it could be
No,
dangerous,
no,
and
no, you're not.
science should jump
Del,
up and be excited to prove me
Del,
wrong.
didn't you get the memo that you're supposed to drink the Kool-Aid?
I know.
Because that's what
I
it
didn't
comes
drink
down
it.
to.
Right.
And me either, because I make my living as a scientist, having an open mind and applying the method of multiple working hypotheses, where I encounter the world and I say, I don't really know much of anything. And anything that I think I know can be changed at a moment's notice based on new data. And that's the stance that I take. I think that's the responsible stance to take as a scientist. And I wouldn't have been successful in the way that I have if I had been one of these true believers that just went with the party line and swallowed that hook, line, and sinker. It's just not how I'm wired. But in this
Well,
case,
thank God you're not wired that way. And it's fascinating
thank
to
you.
hear someone that's as directly involved with the technology that we are talking about. If anyone's going to question it, someone that was there when we started like sort of tinkering with it and playing with it in the way to put it into a vaccine. So obviously you like the idea of it being a vaccine, but there were certain things that would have to be worked out in order to make sure that it was safe. And let me just clarify, just from my own understanding, is it that mRNA technology is dangerous, or is it that the spike that they're using having the mRNA of spike to sort of have the cell express, is that where you think they've really gone wrong? That mRNA could work, but we should be using a less dangerous part of the virus to express in order to create antibodies to stop the virus, not use its major, howitzer that it's attacking the body with. Is that where you're at? That mRNA might be a good idea, but the spike is what's messing up your originally good idea?
It sounds to me like you play slow pitch softball. Thank you for that question.
All right.
So there's a caveat, though,
Okay.
to be, honest and with integrity. The new synthetic lipid and formulation that Peter Cullis and his team at Acuitas at the University of British Columbia have designed, which is what's really enabling for these current generation vaccines. So yay for Peter. He's been working on this for 40 years. I just want to do a shout out. This is another one, many people claim to have done the breakthrough. In my mind, it's Peter Cullis and his team at UBC.
Okay.
But they're using a synthetic lipid, a fat, that's chemically synthesized and engineered to do certain things. And it's never been in humans in large scale. And the data packet from Pfizer that I think you're well aware of, that was released under FOIA from the Japanese regulatory agency, shows that this particular lipid accumulates at high about 20% of injected into rodent model ends up in the ovary. That's very odd. So do I know that the delivery package, if you think of this as a FedEx package, and the RNA is the paper, the memo that's inside the package, and the cardboard part outside of it would be the lipid envelope that delivers it into the cell. We don't know for sure what the effects are going to be of the lipid delivery package. So when you say to me, are you sure that this is going to be safe? The general concept, yes. This specific compound, to be determined. Now, we are completely aligned, and I've blogged that I've had a lot of positive feedback. Frankly, since that podcast came out, I'm having physicians and scientists from all over the world contact me and basically say, yes, go Robert, because I'm able to have this platform, and there's a lot of enthusiasm that what I'm expressing is accurate, correct, and a lot of people are frustrated they haven't been able to get the message out. I think a lot of us agree that these first generation mRNA vaccines based on spike, understandable that they went for the easy thing off the shelf right off the bat. But now we know more, and it's really time to start rolling out second generation products that, for instance, might just express and display the receptor binding domain without the active parts of the molecule of spike, might express matrix protein, might express some of the other structural and non-structural proteins. because those will also all get processed and displayed on the surface of the cell and elicit cytotoxic T lymphocytes. So there's ways that we can go forward that are next generation.
Hmm.
We do have to address, I think, to be responsible, we need to disclose to the public completely what these potential risks are. I think that's fundamental bedrock bioethics. And the public needs to understand these pharmacodistribution studies that were done and needs to be aware that the full reproductive toxicology package was not performed before they started being administered. And that means that we don't really know what the reproductive effects might be. And even if they cleared the reprotox GLP studies that weren't done, and they were all okay, those are in rodents. And those of us that have done animal model work, including myself, are well aware. What we all tell each other is mice lie, monkeys mislead, and the only thing that predicts an activity in humans is humans.
Right.
And now we're at a stage where we're accumulating human data, but human reproductive toxicology data is a lagging indicator. That's going to take a long time to get out.
Yeah.
And the government isn't being transparent about this, but you can talk to 20 different women that have had vaccines. And I'm guaranteed that a lot of them are going to tell you that they've had disruptions of their menses, really heavy flow, other things like that. So that talks about an interaction with a vital reproductive
Well, we
organ.
just at the beginning of this show, we talked about a very interesting study that looked at the spontaneous abortion, and the study sets out to show that the vaccine didn't cause spontaneous abortion, that when they went, it was right about at the background level. But then a nonprofit, not ours, or another group investigated and said, look what they did. Spontaneous abortion happens, I think, within the first 20 weeks or something like that. But they took women that were taking the vaccine in the third trimester and added them to the group of pregnant women. When you take them away because they're not correct, they will never have a spontaneous abortion because they're too far into their pregnancy. We saw an incredibly high amount of spontaneous abortion. I believe it's somewhere in the 70 to 80% range, which is shocking. So, and a lot of these studies are coming out. Let me ask you very specifically, because I could talk to you all day. This is so fascinating. But
yeah, this is fun.
you reached out to the FDA. You said, look, there are issues. This thing appears that it could cross the blood-brain barrier. It could, and you're a scientist, right? You're saying these are theories, but the potential is there. And before we go ahead and launch this thing on every human being on the planet, we really need to rule out these fairly obvious theoretical potential problems. Now, you said there should be an informed consent, which is the name of my nonprofit, Informed Consent Action Network, informed consent to consumers to know there are risks that we have that may or may not exist. But let me ask you, as you started seeing blood clots being announced, first with Johnson & Johnson here with AstraZeneca in Europe, so it mirrored that same technology was having the same problem. When we looked at the VAERS reports and the different, capture systems, we see that Pfizer and Moderna were having issues with blood clots and thrombocytopenia at the same time, which gave it this very unique footprint. They denied, right?
Which the FDA denied in their press release just recently that they gave to PolitiFact. So I received this paper that I mentioned to you from some scientists in Singapore that's just published in a very high impact journal, American Journal of Hematology,
Okay.
in which they report a series of cerebral vascular coagulopathy, so blood clots in the brain. That's kind of not a good thing.
No.
No, I don't want one. Neither do
No.
you.
No.
That kind of takes away what makes you human
Right.
sometimes. So this paper is now published, and it clearly documents those relationships with RNA vaccination and clearly and unequivocally ascribes it to spike that's expressed from the vaccine. And then we have this Harvard and Brigham Women's nurse study in which, or I shouldn't say, it's Brigham Women's and Children's is the institution. So it's a nurse's study that I'm sure, familiar with that shows that we're having free spike in non-trivial levels circulating in the blood after vaccination for up to almost a month in some nurses. And so it's kind of another one of these where there's smoke, um, and it's kind of looking like we got a fire maybe, and we sure ought to take it seriously. I think that's where you and I are very much aligned.
Yeah.
Is in the position that we're not saying that the vaccines don't save lives. My position is I can't say whether the risk benefit ratio makes sense or not, because I don't think they're following protocol and actually calculating the risk benefit ratio, number one. And number two, that idea of risk benefit ratio is typically applied as a function of special populations. So risk benefit for vaccination for pregnant women, risk benefit for adolescents, elderly, all those things have to be
Most of
calculated
which
and
weren't
then a
in the phase
decision
3
made.
trials to begin with, right? Most, like,
Yeah,
we skipped
yeah.
all of
They
these
don't have the data
susceptible
even.
populations, right?
Precisely. So this kind of hand waving, move on. There's nothing here to see. And I, you know, I call it fake it till you make it. Is kind of the way that things are going here, and that's not okay. We got human lives at stake, and their livelihoods and their health and well-being. And there seems to be an overlap between many of these symptoms and some of the spectrum of symptoms associated with COVID and long COVID. And that's a little alarming, and it suggests that spike may be the bad actor here. I'd like to say something else about the ad vectors versus the mRNA. One of the things about ad vector gene therapy technology is it was designed to provide long-term expression of the transgene. That's fancy words saying it makes a lot of protein for a long period of time.
Mm.
How much protein, right? If for the Novavax, just to take a stalking horse, for the Novavax vaccine, which is a more traditional vaccine, the FDA requires that they very precisely calculate the dose, the amount of spike antigen in the formulation that is in the jab that you get in your arm. So you know precisely how much protein is there. In the case of the gene transfer based products, there is no assessment of what the levels or duration of expression are in tissue. Now, technically, the active drug product, I'm using regulatory speak now, the active drug product is the protein.
Right.
That is the thing that causes the effect. And in their wisdom, the FDA has decided not to require that the pharmaceutical companies characterize exactly how much spike protein is being made. We don't know. And it is reasonable to assume, based on everything I know in gene therapy for decades, that the adenoviral vectors may be expressing significantly more protein for a longer duration than the mRNA. So
Okay.
no surprise that the safety signal for coagulopathy pops up first with the advectored vaccines. That doesn't mean it's not going to happen with the RNA vaccines. And we have this huge difference in dose between Moderna and Pfizer, for example.
Mm-hmm.
And then recently we had the press release from CureVac about their much lower dose, which only provided about 40% protection, which frankly is not trivial.
No.
Often we have flu vaccines that only provide 40% protection. Okay, so CureVac seems to have got it a little low. Pfizer might be in the right range. Moderna is way high. And because I work in the government, I have good contacts with the people that were on the team for OWS that designed and expedited the Moderna product. So I know from first-person accounts how that dose was selected. And
Right.
this is another example of fake it till you make it. It was decision making by committee, and there was part of the committee that thought they ought to go with 200 micrograms, and there was part of the committee that thought that they should go with 50 micrograms. They were probably the ones that got it right. And they split the decision because they didn't have solid clinical data to inform that. So that's how that dose came about. Now that we
Wow.
see, and right, precisely, that's how things rolled
Literally
during
rolling the dice. I mean, it's
OWS
just short of,
with people's
rolling
lives.
dice. Yes.
Yeah. No, it's decision making by committee. It's worse than that. It's groupthink. It is
Yeah.
groupthink. If you look up the definition of groupthink, That's what we've been dealing with all the way through this, is a bunch of people who are very convinced about how smart they are, and they want to come to consensus with each other, and they kind of follow the leader, and they all want to get on the same page, and they don't challenge each other like you are challenging them.
Hmm.
I mean, no wonder you're an enemy right now, and you're treated as one, and I am to some extent also, is because we're committing the sin of saying, yeah, but did you think
Right.
about this problem? Okay.
So
And that's our sin.
let me just ask you then, because I want to jump onto one topic. We've already gone, longer than I had promised you, and I hope I can hold you for a few more minutes. But very quickly, if we were going to inform citizens that are stepping forward to get this vaccine, what potential risks should they be aware of if you were going to come up with a list? I've had my own, but what would you say are the potentials that you're looking at that you would like to see a long-term safety trial rule out? What should we know are potential risks right now at the top of the list of these vaccines, COVID
Okay.
vaccines?
So, in terms of the current AE spectrum, um, I I understand that in addition to the cardiotoxicity or cardiomyopathy, and by the way, I think that that is likely to be a broader problem than just adolescents. I think
Okay.
it's just harder to detect because of this problem with statistics that we call masking
Yep.
and confounding variables, because people that aren't adolescents have a baseline level of cardiovascular events that makes it harder to pick out the signal. And you're sophisticated enough, I suspect you understand exactly what I'm saying.
Yeah, kids don't have
So
heart
there's a good
attacks, so when they
chance,
start having heart attacks, you notice it. Adults do, and so when they have
yeah,
heart attacks, they say it's because they were old, not because the vaccine they got
perfect.
4 days ago.
Yeah, and so
Right.
to that point, it's a great case study because to that point, if you're a person sitting at CDC evaluating the safety database,
Yeah.
whether it's vSafe or VAERS, and everybody around you is telling these are safe vaccines, and you see an MI event 2 days after vaccination, just to take an example,
Yeah.
okay, you're going to be biased to saying unrelated because old people have heart attacks, and that's not how it's supposed to work. Just like the other example, it's kind of you're supposed to presume guilty until proven innocent.
Right.
Um, and so I think that that's part of this underreporting, low detection of the signal is the bias on the people that are evaluating the data because of all the messaging they're getting from their supervisors and all the way up the chain in the
Right.
HHS system. Okay, so you asked me
You have
about
the President
risks.
of the
I
United
think there's a
States telling the
chance.
world this is a safe product. Everybody's got to get it. Kids, get out and get it. The Delta variant is bad. Kids, you need it. You're in danger. When the President of the United States is pumping it, Tony Fauci's pumping it, every hospital's being told, get your numbers up. In fact, force your nurses and your doctors to get it. It's really hard under that
Okay,
environment to
you're
say,
gonna go there.
yoo-hoo, I have a problem. There may be a problem.
So I got to say, if you're gonna go there, I got to say something else.
Okay.
Uh, with the censorship, like the Facebook group for people that claim to have vaccine adverse events.
Mm-hmm.
Um, these people that have have had these symptoms, these events, and it's really disrupted their lives. They call me; they're full of tears, and agony and consternation. Um, putting out the message to everybody around them, their family, friends, kids, whatever, that they couldn't possibly have had a vaccine related adverse event because these vaccines are safe
Right.
is one of the most profound gaslighting things I've ever heard of. And then to delete the Facebook group that these people, 250,000, I think, of them have joined,
Yeah.
and then you just delete it and flush them. What you're telling these people is that they're crazy. It is the ultimate gaslight. Okay. And some of these people are committing suicide. They're desperate.
Right.
It is it is I don't want to say a crime, but this is profoundly wrong. Okay, so there's that. Going on to the adverse events. Um, the, so there's another safety signal that has been detected but it hasn't been disclosed, as I understand it, which is reactivation of latent viruses. So this is the
Okay.
shingles.
Yep.
The thrombocytopenia seems to be happening. Obviously, the coagulopathy seems to be happening. Whether or not that's related to the cardiotoxicity signal. By the way, I was the guy on point for DoD to look into the cardiotoxicity signal on the myocarditis that we found with the smallpox vaccines when we started to deploy them. And
Okay.
there's the same problem there. Okay. So,
Interesting.
is that you asked about long term.
Yeah.
I'm still not sure that there isn't some ADE going on, and it's hard to pick out ADE unless it's florid. If you have people dropping like flies after vaccination when they get infected, that's obvious. The
Right.
dengue situation was pretty overt.
Right.
But that's not happening, yay.
Yep.
But there are some odd signals in national health data about mortality, all-cause mortality event rates after a vaccination campaign, after a surge in
Yeah.
vaccination, going up.
We see it
That's
all
kind of
around the world,
paradoxical.
all around the world. As that
Yeah.
vaccine really kicks in, we see a rise in death. Then it goes down, but you really probably killed off everybody that would have been weak to the virus or the vaccine,
Or
potentially.
I don't want to go there and
Okay.
say we've killed off everybody. That's kind of alarmist. But there is some there. I think that epidemiologists
Yeah.
may well pull out data suggesting that there may be some ADE effect. We don't know yet, but I'm still not convinced there's none.
Okay.
It's not florid, which is what I was
Okay.
originally worried about.
Yeah.
Then there's in terms that you mentioned the long terms, obviously the reproductive toxicology, we just don't know. The
Yep.
CDC looked at pregnancy in an MMWR publication that you probably looked at a couple weeks ago.
Mm-hmm.
And they asserted that based on the VSafe database, they didn't see evidence of toxicity from the RNA vaccines in pregnancy. But if you look at the final paragraph, it's kind of got the punch. And they say explicitly the VSafe database is flawed, inadequate, grossly underreporting. We need to do a better job in the next outbreak. Therefore, we're not really sure about whether or not they're safe in pregnancy. That's the bottom line in the CDC's
And
assessment based
VSafe
on the
being sort
VSafe
of that
database.
VSafe being the middleman that's now been put in on the way to VAERS. On the way to reporting to VAERS, VSafe sort of has gotten involved, is helping people through it or whatever. VAERS being underreported, famous Harvard study that showed that it captures, less than 1% of the total amount of injuries. We now are seeing, I think, in the neighborhood of 6,000 deaths being reported at VAERS by this COVID vaccine. If that's an underreporting, even at those numbers, yes, that's where we're at, 5,888 right now. There's obviously a backlog, but that would be the highest death rate, not only of any vaccine that's ever been made, but literally of all the vaccines that have ever been made over the last several years put together have never killed that many people.
Yeah, so that analysis, which is Steve's analysis also, has brought out the haters and those nattering nabobs that pick out various reasons why you can overanalyze that. You can overstate the importance of that data.
Yep.
I'm with you and Steve that we can all agree those are imperfect data, the death. And by
Yep.
the way, there's like 30,000 in Europe. Um, uh, and that those aren't verified deaths, they're vaccine associated deaths.
Right.
Okay. So, but the point that you make and Steve makes is that we have this prior baseline of death events for all vaccines combined up until we kick in with this vaccine campaign, which
Mm-hmm.
means the denominator now is bigger because we've got all vaccines plus this vaccine over that time period.
Yep.
So you'd expect, you might have to do some correction, but the difference is so large and so striking. I don't think how you can look at that data and not say, we better look closer at this. This is a little worrying. So that's why I when I look at those data, I kind of come away. I'm a little different from how Steve looks at it. He's like the house is on fire, and they're killing people. I prefer to take the stance. This is concerning, and it absolutely merits a rigorous follow up so that we can really figure out what's going on here.
Let me be perfectly
So
clear about where I'm at with it, because it'll sort of lead to where I sort of want to sort of sum up this conversation right here, which is this. we are having Facebook, as you said, censorship, that you have people naturally on their own putting up Facebook pages saying, I've been injured by this thing. Have you had anything like this? And it ends up having hundreds of thousands of people saying, yes, here's my injury. In fact, Medscape just had doctors weighing in, only doctors who report, do you have concerns about the vaccine? And most of them talked about their own injuries, saying, yeah, I'm having these issues, and a bunch of negative comments. I mean, as far as you could, see based on these vaccines. Here's my issue. We are being told that this thing has not cut any corners, that we went through a phase 3 trial. It appears to me, and I think you mentioned this before, that we're treating this, though, as we're phase 4, which is we're in the population, when the truth is, if that is the case, if you are in a population study, you better have the mechanisms to study that population for every injury that's happening. And when you cannot answer to the 5,000, if you can't tell us, is it 5,000? Is it 1,000? Is it none? Is it 500,000 based on Harvard's, and you cannot give us a solid number, then you are it's a dangerous trial being done recklessly on innocent people. And so my issue is this: why can't the FDA tell us how many people have died? Why can't they project how many will die? I mean, these are the things, these are why we do trials, and none of this is known, but you have a better relationship with the FDA than I do. Have you reached out to them?
And the
Have you reached out to the FDA?
the reason they can't do it is because they had the option to require the pharmaceutical companies to acquire that data more rigorously under EUA. They had the legislative latitude to do so, and they elected to do nothing. Okay, so you're dead on. If we're going to point a finger at somebody, and we'll see how long Janet stays. I think it might not be too long, but it's not just Janet. There's a culture there. And we could talk about other stories. Absolutely, the FDA has been subject to administrative pressure from above within HHS, and they are not acting independent of other agencies within the government. They I think that a very strong case can be made, and I could relate personal experience that the FDA is no longer independent of political pressure, and that is a big problem. So I think your kind of network angst coming out here, referring to the movie, is warranted. The question is, where do we go from here?
What does the FDA say when you work with them, you work with the Department of Defense. I don't know if we're gonna have time to get into the other part of the topic I wanted to, which is you were really been trying to pioneer working with, drugs, repurposing drugs, and maybe we'll touch on it very quickly. But in that conversation, have you reached out to the FDA and asked them how well is the data collection going? Is the FDA, are they stressed right now, or are they like, we're cool, man, we're meditating here, kumbaya, everything's looking really good?
I have asked the question directly, and what I'm told is that CDC is overwhelmed. FDA is overwhelmed. The CDC is not allowing transparency to be safe even within the government.
Wow.
And that the FDA, what's happened, I infer from what I hear from my colleagues, is that you have a very kludgy historic system, and people that are deeply dug in very much old school approaches to data analytics on the epidemiology in these databases, and are unwilling to consider new technologies and approaches, statistically and otherwise. And we have these very kludgy antique database structures, and they are not able to efficiently process and sort out signal from noise, and they are lagging, and that is a significant problem. And FDA leadership, I know there's significant frustration that current FDA leadership has been very resistant to implementing more modern approaches to data analysis. I mean, we sit bloody Silicon Valley. I mean, Oracle. We have some of the top people in the world, and it's like the FDA are sticking their heads in the sand and saying, no, we still want to work with COBOL, and do things the way that we did it back in the 70s.
Right.
And they have now been hammered with the biggest crisis they've ever faced, any of us have ever faced in public health. And their systems are not able to support the strain, and they've been overwhelmed, and they've gone into their foxholes. And so we see public messaging where people like Tony admits it, Dr. Fauci, he has relied on his opinion and statements that he thinks would be helpful. It's in the Washington Post FOIA emails. I mean, it's quite obvious. He admits to his biases, and then he kind of wraps himself in the cloak.
He did say last week that any attack on him is an attack on science. So he also sees himself
I
as
was just going to
science
say
itself.
that he's wrapped himself in the cloak. I am science. I mean,
Right.
that's kind of a little
Scary.
over the top.
Yeah.
Yeah, it's like he might have lost perspective a little bit, but hey, he's 80 years old.
Right.
Cut him some slack.
All right, really quickly, because I can't let it go, because the work that you do, you're a consultant, you work with Department of Defense. One of the major things that you've been trying to do is work on repurposing drugs. I mean, this has been a huge conversation, because as this vaccine has been promoted from the beginning, other products like treatments like hydroxychloroquine, ivermectin, is your opinion that there have been treatments available throughout this process that could have curbed the death rate that we're seeing, hundreds of thousands dying? I guess we're in the sixes now in America.
Absolutely.
Could that have been shifted had we used drugs that were maybe pennies on the dollar
I
and
feel very
not
strongly.
patented?
And so, yeah, ivermectin is one example, and the data are coming in fairly strong that ivermectin has benefits both as a prophylactic as a therapeutic for severe disease and hospitalization. The argument that many of those studies, often in emerging markets, because they're very poor and they are using this off-patent, readily available drug that often they have available because there may be endemic worm diseases. Yes, so I was very involved in getting this thing processed and published, this paper from Pierre. And I had recommended that we use ivermectin in one of the arms of our outpatient study with DoD that we're currently prosecuting. Oddly, the FDA required that we prove the mechanism of action of ivermectin using cell culture, which is over the top, if they would allow us to proceed with clinical trials. They did not insist on that in the clinical trial that the NIH is launching, Active 6, testing ivermectin in outpatient. I don't know what to say about that.
So they basically, let me just very quickly, they want you to go all the way back and prove that ivermectin does what you say it does, even though we have like hundreds, if not thousands, trials around the world showing it's working on patients. Can we do one in the NIH where we have a control group and say, we're not even going to do that. You got to go back 12 months we want you to waste in a cell culture study just to tell us that this should be an important study that we should try it, at which point it's obviously too late. We've all gotten a vaccine that is or is not dangerous to us.
Yeah, I like to say, yeah, but 500, 1,000 are dying a day here in the United States,
right.
And you're worried about the potential risk of a 40-year-old drug that has one of the best safety records in the world, administered at known safe doses. They're basically the same doses, by the way, that I administer by kilogram to my horses.
Right.
So it just seems there are a bunch of things here that don't make sense. And by the way, we barely scratched some of the issues. We haven't talked about the lab leak, what went on in Wuhan, what the documentation is about when they actually started building the vaccine. And the repurposed drugs there's going to be a good book about that. For our studies, the FDA is still throwing up just objection after objection after objection. We, each time we come in with our IND for the two drugs, I'm not pumping our drugs. I'm not going to go there. but that we've identified. The trial, we have an IND that we're trying to prosecute with the agency, and colleagues in India are probably going to start a trial fairly soon. But with the FDA, it's a paradox. Every time we have a meeting with them, they say, "Well, you've got to do these things, and then we'll let you go." And we say, "Okay, we'll do those things." Like the ivermectin, they objected to, so he said, "Okay, we're going to drop the ivermectin." DOD just didn't want to have that fight with the FDA. So this happens again and again and again. And each time they come up with new objections. It's very odd. The people on my team and myself have never seen anything like this before. And I don't wish to be a conspiracy theorist, but it just doesn't make sense.
But it's not even consistent, right? I mean, there's no consistency because on the one hand, the very agencies that are rushing out a brand new technology that's going to have free-floating spike all through your body, no long-term understanding of the safety of that is being promoted by the President of the United States, are making you jump through hoops for a product that's been proven to be safe for 40 years.
Are blocking the development of known safe agents that are off patent, that drug companies can hardly make a nickel on, that are, instead of remdesivir costing $8,000 a treatment, would cost something like they're being used in hospitals, these agents that we're working with, and the treatment costs about $50 for a hospitalization, and we pop them out and they appear to have better efficacy than remdesivir. Now, something's not right there, and I think most of us can put a name to it. And no, this is another one of those, it looks like a duck, walks like a duck, and quacks like a duck, but we can't call it a duck.
So name that duck.
But that duck is regulatory capture by the pharmaceutical industry.
Yeah.
Yeah.
Meaning, the very agencies
Janet, I don't think Janet's going to be around very long, but I like to say if there's a wiki page for regulatory capture coming off the Purdue Pharma OxyContin situation, probably going to have her picture there.
What is your advice in wrapping this up? Children are now, we have college students right now. They're being told by universities in the next 2 weeks they need to be vaccinated or show their vaccine in order to go to college this upcoming semester. You work closely with regulatory agencies. What should really be the statement to people who are looking their children, at least under these circumstances, or students going to college? Should they wait? Should they hold on a second and try? I mean, we haven't even had the CDC weigh in on really the safety of this issue of blood clots, thrombocytopenia in youth, and the myocarditis. If they're not weighing in, should we have a pause right now before we force young people that really are at almost no risk at all from dying from this virus?
So that's number one. That gets to the bioethics of the age of informed consent. And so absolutely, unequivocally, people lower than the age of 18 should not be coerced to receive vaccine. And if they do receive vaccine, there has to be parental approval. That's just the rules. The WHO modified its webpage yesterday or the day before. And you're probably aware of that. And there's a new clause. I don't want to get trapped in getting the words wrong and having, a fact check from PolitiFact.
Okay.
But there is a clear and unambiguous statement that the WHO believes that most of these vaccines are safe down to the age of 18. And there's a statement that there should not be mandated childhood vaccination. So the implication is they're defining childhood as including adolescence.
Okay.
So we now have the WHO staking out a position. I was asked by a podcaster in the UK, point blank, do I think children should be vaccinated? And my personal opinion is no. The justification that Delta variant is going to spread more readily and creates an added threat to children. My problem with that is it sounds an awful lot like scare tactics because I haven't seen any data on the effects of the Delta variant in children. And so if you're going to make public health statements, they ought to be data-based. That's the core here, is let's do science-based medicine. Let's stop the fake it till you make it. So if Delta variant represents an enhanced risk for children and adolescents, show me the data. Show all of us the data. You owe it to us. My
Right.
point is that the government doesn't have the right to coerce you to do things with your body. It's their obligation to convince us based on real information about and give us the information that we can make our own judgments as adults because we're competent. We understand intuitively risk benefit. We know that some people die in airplanes, and yet we take air flights all the time, likewise with our cars. We could process this stuff. We're not children. We don't need the government to tell us what to do. What we need the government to do is be frank and open and honest about what the actual data are and stop the scare tactics. So, where I come down is children and adolescents. I'm not aware of data about the Delta variant actually being an enhanced risk. It may be. Show me the data.
Right.
Otherwise, all the information that I've seen is the risk of COVID disease, which is not the same as infection,
Mm-hmm.
in adolescents, children, and infants is remarkably low. And the risk of adverse events in those populations is not nothing, but not nothing divided by almost nothing is a big number. It's not the right kind of risk benefit ratio that normally would be used to justify a vaccine intervention. So I I I think that that ratio has too much risk, too little benefit, and if the public health service is going to make a case that they should be vaccinated, let's see the data. Let's see the actual calculations. In terms of young adults being required to be vaccinated before they return to their universities, I participated in a town hall meeting at the University of Delaware with some very aggravated parents and young adults that go to school there that are being mandated to take these experimental vaccines. That sounds like coercion to me.
Mm-hmm.
And that's not right because these are experimental products. And I suspect that those university administrators who are probably making some sort of financial risk calculation about the lawsuits they might get from COVID infection on their campuses might want to think twice because they ought to factor in their risk benefit calculation for their financial risk, the potential lawsuits they're going to get from people that have been denied the opportunity for higher education because they have elected to not take vaccine.
Right.
Have I addressed your questions?
Yes. My final question is this. You quietly work with the FDA, you work with the Department of Defense, you are in the mix. Unlike many people that sit there as doctors, they have no actual action or way to have conversations with people that might matter. You've been having those conversations. What led you to leave just having those conversations and then 2 weeks ago be involved in a podcast that has now made you a public figure in this discussion? As you said, your phone is ringing off the hook. That's a pretty big shift from someone that doesn't want to be in the media, that's just trying to address the issue, to someone that is now saying things like you're saying, the FDA is overwhelmed, the CDC is overwhelmed, they are not collecting data properly, and this thing is being moved forward. What made
So
you
the
do
pivot,
that?
the seminal event for me was when Trial Site News asked me to evaluate the Pfizer data package and the European Medicines Agency assessment letter that they publish normally. So the one was FOIA, the other is normal disclosure. And I encountered and I worked through those, and it was clear to me that this wasn't right. And it was not right and proper. And then there was the Dr. Bridle response and assessment from Canada. I was on a phone call with the Canadian physician that told me about what he was experiencing and how the government was just summarily dismissing his claim, his posting to the government database of adverse events that his patients were encountering. And he told me about how the government and there's a whole cadre of self-appointed academic police that attack physicians and scientists that say anything about this. I don't get that, except for I was in academia and I know how that culture works, and it's a little twisted. But I saw all these things coming together. I saw the suppression of information and communication. I saw the safety signals. I saw the misrepresentation about how the vaccine had been characterized in the Pfizer case. I saw the New England, the Harvard and Brigham and Women's nurses study, and it just came to me that we'd reached a tipping point, and that somebody, that, and then the whole RNA inventor thing became a big kerfuffle, and I suddenly found myself in a position where people were looking to me um, to provide leadership, and I was being able to say things on LinkedIn and social media that other people were being censored for. And I think it was because of my CV and these past contributions. And people, perhaps the censors, felt that I had enough legitimacy that they were going to think twice before they just automatically whacked me. So I had a platform, I had the CV, legitimacy, the signal was there. There's a bunch of worrying things. I'm seeing my fellow physicians just being hammered if they even start talking about it. And so I started speaking up, and one thing led to another. And there was the invitation to go talk to Brett, and we had an open discussion that some people find is not boring for 3 hours, if nothing else, because of Steve talking over me for most of it, I think. But that seemed to go pretty well. And then people started inviting. They wanted to hear what I had to say, like
Yeah.
you. And so I'm grateful for that. And now it's snowballed, and I've got almost certainly 35,000 Twitter followers. And I've got docs and scientists from all over the world coming to me and saying, "Hey, can you make it clear to people that this is happening, or this paper, or that finding?" And so now I'm in a position, uh, ethically almost, where I feel the need to use this moment in time and this platform to share key information. I think another one was after that Canadian phone call, I woke up the next morning and had the insight about how wrong this was from a bioethics standpoint. And I wrote a piece in Trial Site News where I laid that out. And for me, that was kind of if there's a red pill, people overuse that metaphor, from the
Hmm.
Matrix.
Yep.
It was that bioethics argument and the things surrounding it, which, as you say, is entirely aligned with your nonprofit. So
Yep.
congratulations on that.
Thank you.
We are of one mind. Bioethics should not be violated. Let's not have another Tuskegee. Let's not have more torture. Let's not have Japanese internment. Crises like this, in my opinion, sometimes lead governments and cultures to cross lines that they said they wouldn't cross, and they almost always end up regretting it. And I've been through too many outbreaks. I know how frightening it could be. I can't tell you how frightening it was to get a call from the Pentagon as I was on the road to Ames, Iowa, that said, "Robert, we've just come out of a briefing, and there's a scenario of 1 billion people dead from Ebola, and you've got to do something. How'd you like to be on point for that?"
Yeah.
Um, talk about save the world, right? Robert,
Right.
you got to figure out some way to make a heck of a lot of Ebola vaccine, and you got to do it now, because there could be a lot of people dying. Could be your mother or your wife or
Yeah.
God only knows what else. And so I've been there in the jaws of that pressure, and I understand how hard it is to make decisions in that environment. But I think that the dust has settled. We have a little bit of oxygen right now before Delta hits us. I don't know what that's going to do to the
Yeah.
states. And so let's take a good breath, look at what we're doing here, and take stock, and let's try to get back on track ethically and organizationally. Again, I'm going to say it again. Let's stop the fake it till you make it stuff. Let's go. Let's do science-based medicine, please.
Amen.
So I'll leave you with that.
From your lips to God's ears. Thank you so much
Just
for
so.
your time. Thank you for being brave enough to speak out, a man in your position. We live in a time where they so rarely do, which I think could lead to the death of science. So I think that we are fighting, for science right now at the High Wire. I believe you're doing the same. I believe in science. I know how science is supposed to be done. That is not what we've seen over the last year and a half. We need to get back to it, or it could be very, very damaging for the future of our species and certainly of our education of our children. So I want to thank you for the work that you're doing and bless you on your way as you continue to speak on all the platforms that will have you. So thank you for taking the time and joining us today. If you liked that clip, then be sure to check out our live broadcast of The High Wire every Thursday morning at 11 a.m. Pacific Time. You can watch it on Facebook, YouTube, iTunes, and Twitter. We'll see you there.